Well, good afternoon, everyone. Welcome to the 2013 COVI Lecture Series. I'm hosting today. I'm Dr. Jim Wilworth, viticulturist here at COVI. We're in our 6th year now of the annual COVI Lecture Series. And today I'm very, very excited to introduce our first speaker of 2013. It's a real pleasure to introduce my friend and colleague, Dr. Kevin Kerr. Dr. Kerr is the principal consultant and founder of KCMS Applied Research and Consulting, and he's also a COVI professional affiliate and research associate here at COVI. He's a nationally and internationally recognized specialist in research and advisory services for the grape and tender fruit production industry, and along with IPM for these various crops. Kevin has delivered a lot of seminars across the world, in Canada, throughout Canada, the US, Australia, China, and Australia. Or New Zealand, I should say. I said Australia twice. Must be interesting. He has authored a lot of different fact sheets, done a lot of grower articles, workbooks, and seminars for government agencies as in the academic community and through the outreach services here at COVI. He's also a lecturer here at the university and has lectured grapevine biology for the viticulture component of the OAVI program. In terms of his education, he received his Bachelor of Agriculture at the University of Guelph, Masters of Environmental Biology at the University of Guelph, Bachelor of Education, at the University of Western Ontario, and his PhD a few years ago now from here at Brock University. So it's my distinct pleasure to introduce Kevin to give his talk today, Grapevine Nutrition, Healthy Vines Equals Hardy Vines. So I'm really interested to hear what you have to say.
Thanks, Jim. It's nice to be back again. It's kind of funny, and fortunately there's a pile of familiar faces in here from the fall lecture, and your comment was a PhD a few years ago. The line I heard was, if you're ever asked whether it's a good PhD is a done PhD. So when you go into the world of academics, you start to learn these things pretty fast. I'm going to do a lot of recap about vine nutrition. There's been a lot of fact and fallacy going around. And the reason I titled this is because if you're going to have the hardiest possible vine, you have to have a healthy vine. And it does come back to the vine nutrition, and it's not just a singular thing of finding that one silver bullet or that one magic entity. So what I'm going to do is try and go through and get you to maybe change your mindset a little bit of how you decide a vine is healthy or not, but also how you decide whether you're going to add fertilizer or not and what you're going to do with it. Now I'm going to work from a couple of computers because one allows me to see my notes and the other one doesn't. So we're going to work it this way. So what does it have to do with nutrition? Well, we have to think about what are your original objectives. The old line is, if you try and solve every problem at once, you will never solve any problems. So you have to start to look and break them down. The key things that I've always talked about, and I hope that people get from this, is talking about really vineyard production is 3 things. It's got to be environmentally responsible, so you can get proper production to grow grapes each year. It has to be marketable, high-quality grapes each and every year, because if you can't sell them, then you're going to go out of business, so it has to be sustainable. And the third part, you have to understand the practices you're doing, and they have to be done economically. You can't always lose money growing grapes, or you're going to go out of business. So it's something that all 3 things have to be into play before you can really look at it. So when we start to think of things in terms of grapevine nutrition, what am I going to really talk about? What are the critical elements necessary for growth? So what influences the nutrient availability? What do you have? You need to be able to take stock of what's available to you. What's a soil test do? What's a tissue test do? What are some of the things that you can do to avoid some luxury use of fertilizer, or as I call it, making sure that whoever's selling you fertilizer has a really nice vacation because you're giving them all the profits? And then finally, to take off on Letterman's top 10, there's a vine nutrition top 10 things you need to remember at the very end to take it forward. So when you do this, you have to think about, okay, there's no one size fits all. And the reason I kind of picked this slide, one, hockey being Canada's national game, but you would understand that the nutrition demands of Zdeno Chara at 6'8" are going to be a little bit different than Martin St. Louis at 5'7". Even though they're playing the same game and they're trying to do things together, the nutrient demands of each of them are going to be different. So there is no one universal program that's going to work everywhere for every variety under every condition. what influences the nutrient availability right now? Thing to think about is water and nutrients limit vine growth and productivity. So fundamentally it's two things. Either the plant can access the nutrients, but you need water to do that. How do they make them available? Well, you got to have enough precipitation to get soil moisture levels high enough that you can get movement of the elements from the soil particles into the roots and be absorbed. If it's too dry, it doesn't matter. All the elements in the world can be there. It still will not move from the root or from the soil source to the root source. We have to remember, the more evapotranspiration that's taking place, the higher the water demand on the plant, we're going to have to make sure that there's also going to be more there will be more nutrients taken up at the same time. They go part and parcel. They follow concentrations. We have to think about the mineral element balance within the soil, the competition on the space on the soil particles for uptake by the roots, and finally one that gets missed by a lot of people is the fact that each cultivar, each clone, and the combination with rootstocks will have a differing demand and availability of nutrients. A classic example is Merlot. Merlot is a very high demand for potassium. If I put it on a rootstock like Riparia, which has a low ability to uptake potassium, I'm more likely to see potassium deficiency in that vine, even though my soil test may say there's lots. This is where the imbalance comes in, of how we have to think about they come into play. So you can't simplistically look at it in one way or the other in terms of availability. So, the other thing to remember, the bulk of the nutrients taken up by a grapevine are in the top 50 centimeters of the profile. The wonderful stories of grapevine roots going 7, 8 meters into the ground, into the chalk, going deep down into the limestone and everything, all these wonderful things. No, those are structural roots for support. A little bit of moisture uptake. The bulk of your uptake of nutrients is in that top 50 centimeters of your soil profile. And in fact, some even say 80% of the nutrients are within the top 25 centimeters of the profile. So when you think about it, this is where the fine fibrous root system and everything is critically important. The soil parent material will dictate the availability of these nutrients as well. So that's going to be, what do you start with? Is it naturally inherent in that soil? Do you have a limestone base, which is high with calcium and magnesium? Or do you have a high clay base? Do you have high organic matter levels? All of these things will influence each mineral element that might be there. What's your organic matter levels? Are they high or are they low? Part of this is for water holding capacity. Again, going back, if I influence water holding capacity, I influence the soil moisture level, I influence the availability of nutrient to be picked up by the plant. And finally, the last misnomer, or the myth that's out there, vine rooting depths, roots will grow into nutrient-rich areas, but they don't seek out nutrients and water. What I mean by that is if you have a soil profile that's very, very good, and you have a slight hard pan and then really rich soil just slightly below that hard pan, the roots do not know enough to go through the hard pan to get to that soil. They don't have brains. We can't give them human characteristics. They continue to grow in good areas. So by creating a good soil profile with good access, then your root system will get somewhat larger. If you confine it or restrict it either through a hard pan or it could be saturated soil where we have really high elevated soil water levels in the spring. Comment that was made to me years and years ago, it said grapevine roots don't mind water but they don't swim. In other words, a saturated soil, a grapevine root will not grow in. It will not survive. It will stay away from it and perch, and it will stay very high. You need to understand different minerals are available at different depths based on your parent material and what's available to begin with. Finally, is the mineral mobile in the soil in terms of water? Can it be leached away, or is it going to stay and is it going to fix to the soil particle? Similarly, is it mobile or immobile within the grapevine? Some of the elements can get picked up by the roots and move very easily throughout the plant from point to point. In fact, will be redistributed. Others, no, they don't. They're a large ion or an element, and they can't move very far. So there's a very short period of time in which they can become available. And then the last one again, what is my cultivar demand? What is my clone demand? What are the rootstocks and how does it influence the uptake? And what are all the interactions of them? So it's not, I'm starting to get saying, there is no silver bullet because there's way too many different factors that I have to take into account. So generally to solve nutrition problems is not solving one single little element. It's often solving a bigger picture. But you need to know what each element and the rest of them do. So in this, what we're looking at is in the early season, we've got a situation that the first 3 expanded leaves you see in a grapevine are all based on stored reserves in the plant. It has nothing to do with the root system growing, functioning, or accessing mineral elements from the soil. It's based on what happened the previous year. Why is this important? If you send a vine into the winter very stressed with low reserves, it's going to have more difficulty the following year. If you had a great fall for maturation, good reserve accumulations, you have a really bountiful situation where the grapevine can grow very quickly in the spring because it's got a lot of reserves to mobilize. However, further growth requires the nutrients and the minerals, nutrients and water to be taken up. If we have insufficient nutrients, we don't get the growth. If we have water stress, either through drought or through excess water, in other words, a fully saturated soil, growth will be restricted and nutrient demand will drop. Nutrient deficiency, what is exhibited, what you will see most of the time when you think of it, less cell division. If you have stress of nutrient deficiency, this can have an impact on final berry size, especially after bloom. It can have an impact on cluster size. It can have an impact on shoot growth, berry numbers. We know that photosynthesis will be depleted or decreased as a rate. Overall vine health goes down. Our fruit quality generally is poor. A little bit of stress is okay, but true nutrient stress is not. And finally, an unhealthy plant, for any of the above reasons, is a non-hardy plant. You have a challenge going into winter. So coming back to these nutrient levels. Now the flip side is we don't want to become afflicted with what some may often refer to as rich man's disease, where you just basically you eat way too much, you drink too much, you get too big for your body. That's luxury use, and yes, you put yourself in a stress position again. So you really have to find this balance. What are the critical elements in terms of looking at it? Really quite simple. They're referred to as the macro or the major nutrients: nitrogen, phosphorus, potassium, magnesium, calcium, and sulfur. Generally, these are taken in high demand because of function within the plant, but also the size of the element itself. They're generally large elements. The other ones are the micronutrients. These are the ones that have a tendency to act in concert with enzymes. Some of it's part of with photosynthetic activity, some of them is with ATP, ADP, in terms of movement through the cycles. Iron, manganese, boron involve a fruit set, copper, zinc, and chlorine. I'm going to go through a little bit on each one, just so it puts it into play for what you're thinking about. The role of nitrogen. I mean, most of us think of nitrogen, and anybody who has a home yard thinks of, like, throw nitrogen, I got to cut my grass 47 times. Well, that's a pretty good equation. Yes, it's one of the ones that's an essential element. It's part of chlorophyll for photosynthesis. It's part of the genetic building blocks, right? Your DNA. It's part of the enzymes regulating the vine growth. Primarily, it's taken up in a nitrate form, okay, that the vine can use. Sometimes an ammonium form can be taken up, but generally it's preferred not to take it up, and especially during bloom. Ammonium at bloom is a bad thing. What sources of nitrogen can you have in terms of trying to get it going? Green manures, which is natural green growth there, animal manures, commercial fertilizers. In fact, you can get some fixation of nitrogen by the bacteria in the soil, and organic matter, which is broken down by the soil microbes that are finally released. Signs of nitrogen deficiency. Root growth sometimes will increase, but it will relative to shoot growth. So there may be a mild, because it's not being translocated away, it's being accumulated. A secondary thing that can occur is that drought susceptibility increases because we've got a stress on the plant. Shoot growth, yeah, we see shorter shoots, shorter internodes, photosynthesis drops down. An interesting thing is if photosynthesis shuts down and the chlorophyll starts to break down, we have a tendency for sugars to accumulate in the leaves. And that's when you start to see some of the stresses. And you'll see a yellowing of the leaf because, and when that yellowing starts to happen, the other thing that happens if that leaf finally shuts down from nitrogen deficiency, it will discolor and anthocyanins will increase in the leaf. All because it's not being mobilized. And the last one is, in the fall, premature leaf senescence, and there is some nutrient recycling, and that plant will scavenge from older leaves for younger leaves. When we look at phosphorus and where it comes into play, phosphorus has a whole series of things that it comes into play with. When we look at it, it's involved with fruit quality and wine quality at the end. It's been associated somewhat with an example here. This is actually an infection by mildew with disease resistance and health within the plant. And in fact, some phosphorus compounds are being used for disease control as foliar applications.
It's heavily involved with root development and root expansion. And these are just different showing, these were from Terry Bates showing what would happen with C3309 in terms of how the rooting profile. And phosphorus is also involved with the flowering process. It's part of the balance relative to the calyptra drop as well as the actual pollen tube germination. So all of these things come into play, and that's where phosphorus has a big role. Now, if we look at it and look at phosphorus deficiency, what are the signs that we might find with it? Root growth, in fact, roots will be more shallow. They don't have a tendency to go as deep, which increases our drought susceptibility because less water access. Shoot growth goes down, carbohydrates, photosynthesis. The interesting thing I didn't realize, what it can do is reduce the transport of magnesium through the plant. And so you can actually have what appears to be a magnesium deficiency on the leaf that can be traced back to a phosphorus problem at the root level. So that's why sometimes what your eyes see or what a laboratory tells you doesn't tell you all the story. And you need to be aware of it. And again, anything that restricts the leaf activity for photosynthesis is premature leaf senescence in the fall, which is always going to impact on fruit quality at the end of the season. Always. And the last of what I call the big three is potassium. And the reason I call it, they refer to them the big three, you buy a fertilizer bag, it's always nitrogen, phosphorus, and potassium content on the bag when you buy the materials. Well, where does potassium come into play? Potassium has a big role, and it's something to be aware of. It involved very heavily with water uptake from the roots and stomatoconductance of water movement through the plant. So it's a water regulator. It's also involved with water retention in the plant, if there's a situation there. It is involved in carbohydrate movement. What was I found very interesting was that carbohydrate movement into the fruit is highly associated with potassium. Now I'm going to talk about sugars and starches in a bit from the leaves. It's not potassium that's involved with the movement out of the leaves. Nitrogen uptake, cell growth, vine hardiness, all of these things are in play. So when we take a look at them, we try and think. And I put this one in just to show you, even in Ontario, we can have a situation where we can have floodwaters in vineyards. This was taken about 6 years ago at the top end, I guess up about line 6, and all the drains were full. Every ditch was full in June, and that was a new planting, and they literally all their tiles had backed up. Now the plants survived this. Short-term exposure to saturated soil is not a problem, but long-term exposure, yes. When we look at potassium deficiency, the signs there again, root growth is more lateral. Sugar export, what happens there is that if we have a potassium deficiency, what happens during the ripening phase, the plant starts to scavenge from the trunk and the roots to try and find materials to send to the fruit, which is interesting. So now you have an implication relative to vine hardiness and overwintering, because now I'm taking away reserves required for overwintering. It can impact xylem flow, and the other part again with potassium. Sometimes the deficiencies we see is due to the cultivar and clone interaction relative to the yields that are there. So in some cases you actually have to saturate the soil with slightly higher potassium levels so that the roots through osmotic movement will actually get better balance and get more uptake rather than active uptake. Being aware, as I said, Merlot, Dr. Denis Boubals from France was over one time, and how this happens. You always bump into people in a place you never imagined. I'm in a hotel in the Okanagan, and I'm going out to look at some stuff, and I get up for breakfast, and I walk out, and Dr. Boubals is sitting having breakfast. So they asked me to go look at these leaves in this one particular block. I thought, geez, there's somebody who's been around the world more times than I'll ever imagine. And I said, I'm going out to look. They've got Merlot. I said, it's on Merlot Clone 184. I said, it's on one section's on 3309 and one's on SO4. And he looked at me and says, yes, the 3309, those leaves are red, aren't they? I said, yeah. He says, oh, 3309 is not good at getting potassium uptake for Merlot. Okay. So then my question was, well, how do you solve it? So I would look like a hero going out to talk to these people. He says, oh, we just have to add more potassium to the soil. It's just it can't get enough fast enough. But there was a classic example. If you didn't know about the interactions, you would never find out about them. And that's where experience comes into play. And the last one is looking at is magnesium. Part of chlorophyll, we're all aware of that. Aids in the formation of sugars and flavor compounds, big part in the flavor development, enzyme activity, and part of proteins. Where it's important to recognize is in deficiency mode, though, and this is one in Marcus Keller's book I started to scavenge through it. I
anytime I get a new textbook, I kind of try and go to certain spots because hopefully they'll have way more recent information than the stuff when I was an undergrad. A couple of things he mentioned on magnesium deficiency have been published. Photosynthesis. That starch and sugar accumulation or levels in the leaves will be very high if magnesium levels are low. And the leaves become far more light sensitive. That's why they discolor and lighten. And the other part was then they'll darken. Anthocyanins will accumulate. Magnesium is heavily involved with the movement of sugars and starch out of the leaf into the phloem of the rest of the plant. I didn't realize that. But again, a sign. Most of us would look at depressed shoot growth, magnesium. Older leaves are more likely to show a situation with it. And magnesium's been implicated with bunch stem necrosis. Late season, premature shelling of fruit, darkening of the rachis on the clusters. That's where it's been. If you see it late season, it's likely magnesium. However, if you saw a similar symptom during the bloom period with poor fruit set, you might associate that with excessive ammonium or nitrogen uptake. So knowing the time of the year and where and what you're looking at can give you implications of what caused the problem. The micronutrients we talked about in already a little bit. Chlorophyll for iron, manganese, photosynthesis, boron, pollen germination, and fruit set. All of these have to be in place before the plant needs it. Putting it on after the fact is like painting wet wood. Might make you feel good, might look good for a little bit, but it really hasn't corrected the issue. You need to be able to anticipate. An example there with boron, if you've had fruit set issues that you can actually identify and come back to say it's a boron issue, you need boron applications very early in the spring, well before bloom. Not at bloom, not after bloom, prior to bloom. Or very late in the season so the leaves can translocate and keep it in the overwintering buds. Copper, root growth, rarely have I ever run into issues of deficiencies there, similar with zinc and chlorine. You know, this is being pulled from the literature in terms of the impacts that they have. So when we look at it, we want to think about the soil. So how do the nutrients get to the roots? Well, there's an impact of soil pH, soil moisture, plant growth stage, the demands for the mineral elements in the plant. There's different times in which they need them, and finally mobility of the soil and within the vine. Everybody who's taken 2P99 and anybody who's taken any basic soils course anywhere in the world gets to see one version of this slide everywhere. Our ultimate target that we try for grapes is to try and look at around 6.6, 6.5 to neutral, 7.0. And the reason being is that that's where the bulk of the elements are available, all of them equally. The dilemma is if we start to go more alkaline into the 7s, then we start to run into some of our micronutrients become much more limiting, iron, manganese, and boron. Similarly, if we start to get too acidic, then we start to look at maybe some of our macro elements that are becoming less available. So going to excessive in either direction. So fundamentally, you try and target neutral, slightly acid often a little bit better, because it's easier to lime and neutralize an acid soil than it is to acidify an alkaline soil. Far, far easier. It's called lime. We can always add lime quite easily. But to try and find acid formers and put it on a soil that's already high pH is really, really hard. Ask anybody who tries to grow blueberries and wants to get their pH down to, blueberries grow best, actually, believe it or not, around 5 or less. And you got a soil at 7, trying to get it down is next to impossible. You could have everything else right, but that's really, really hard. The other part is soil water relative to soil texture. The key to remember, if we look at it, here's the wilting point. In other words, we have to have enough soil moisture present above that level so that the nutrients are available. This is our available water. If we go above that, generally what happens is it starts to leach out, goes beyond. We can hold more water in clay, but less is available than if you were in a silt loam, which has a higher range of availability. Sand has a very narrow point before it leaks out. Imagine this is an okay sponge. This is the best sponge. This is not quite as good sponge. It's the way to simplify it and look at it. Mobility in the soil and mobility in the plant are not always the same. Key examples: nitrogen, highly mobile in the soil, means it can be leached. Highly mobile in the plant. Well, that's good. Phosphorus, very low mobility in the soil. That means we have to put it where the roots are. But once it's in the plant, it can move all over and be utilized throughout the plant. Potassium, low to medium mobility in the soil, but once we get it in the plant, it can move around quite easily. So taking these into account,
And calcium is a low plus low. Means I got to do everything I can to get calcium in, and in fact, in many cases, calcium additions as a soil application alone, where you have problems in a plant, are generally not sufficient. It's one of the few ones where foliar applications often are much better to correct problems. Magnesium has low mobility in the soil, but high mobility within the plant, and sulfur has a medium and high. The micronutrients, when we look at it, generally, I was quite surprised to find that they had somewhat low mobility, but part of it can be back to the element form. Zinc is not very mobile. Boron is highly mobile in the soil, but not very mobile in the plant. Boron is a real one that I would recommend to watch because you can move into boron toxicity very easily. And a symptom of boron toxicity is identical to boron deficiency. And many laboratories will give you high rates of boron applications thinking you've got a boron problem. You don't. It's probably one of the most dangerous ones around. So why do people use fertilizers? I asked the question one time. Well, they do it out of habit. Every year in this block, I'm going to go out and do it. Now, it could be a good habit or a bad habit, I don't know. Second part is there's a good salesman running around the area telling you, you really need this is the best thing you can buy, and they get all kinds of people to buy it that year, and nothing changes, but did a great job of it. Or the other one was, and this one I like, it's two little kids with what I would call a muskrat trap, and we got a bear. Well, this is just wishful thinking. They think that by throwing fertilizer out there, it's going to do what it's supposed to without ever really understanding what's really necessary to do the job. Either put the right amount there or don't, because if you're trying to trap a bear in a muskrat trap, it's not going to work. And finally, the last one is, well, the lab told me to. Well, the laboratories are very interesting in that laboratories work on bulk numbers of samples and bulk averages, but they don't take into account a lot of things when you send the sample in. They don't know the age of the plant properly. They don't know the crop load on the plant. They don't know the health of the plant. They don't know the intention of the training system that you're on. They don't know whether you took a correct sample or not. All they're going to do is say, I can analyze what you gave me, and this is what I think you need. The other part that I learned is that the laboratories base recommendations on their parent material where they're generally located. So if you have a laboratory in Ontario, yes, they will work from Ontario conditions. But if you send it to Michigan, if you send it to Ohio, if you send it to Iowa, they will base it on the parent soils of that area and what they think it should be. Very different. Very, very different. In fact, because their parent materials don't have a lot of it, sometimes they will say, oh, you need to overdose on things to try and supplement something that's not naturally occurring in their soils, but we already have. So you have to really watch. So how do you decide? Well, this was the one I was trying to go, you gotta change your mode of operation the way you do this, and it's one of the hardest things to do. You've got to observe the patient in the field. Go look at your vines. Walk around. Don't rely on what somebody else tells you that they are. You've got to understand the lifestyle. If you went to your physician, they want to know how you live, what's the crop load, what's the pruning, what's the training, what's the environment that they're in. You want to check the patient's diet. This is where the soil test comes in. So it's only one part of the equation, the soil test. And then you want to physically check over the patient. Are the shoots really growing? Do you know that they're growing well? Are the shoots growing? And you can do this through a tissue sample. It's no different than a human being going through for blood work, right? They want to see the patient. They want the blood from that patient to understand what that patient's all about. Then they'll work in generalities to come back. But not everybody's the same. And you have to remember that. So soil and tissue sampling, what do? They give you base levels. They can diagnose problem areas. You can look in a spot and say, something's wrong here. I want to compare that area to another area on my farm. You can monitor what's going on, whether there's been changes over time. You can come up with your fertilizer and lime requirements. So, why do people take them? Again, I asked some people that I work with. I said, well, why would you go? First off, they would say, you know what? I got a poorly performing area. It's not growing as well, or the fruit that's coming out of there is not as good a quality as I want, or I'm not reaching the standards of fruit quality that I'd like to. So is it a nutrient issue? Is it something restricting in my plant that's causing an issue? Or they have just poor yields. They're not getting good fruit set, they're not getting regular fruit set, something's going on. Or they're saying, look, I want to try and push the plants a little higher. I need more crop production. If I'm going to be economically viable and I'm growing into a certain category, I can't make a living at 2.5 tons to the acre. It's going to go into this group and I've got to grow it at 4 or 5 tons per acre. Can it be done? Can I assist that? Another one that's been done for people that take it over time, that can correct the issue before it becomes a problem. So you watch for trends. Rarely will something become a serious issue in a single year. It occurs over a period of time. So the idea is following it. If you see a steady trend downwards of certain results, then it's going to tell you that's likely to continue to decline, and you may get below critical levels. The other part is, finally, for those that take them on a regular basis, they understand that their vineyard is different than everybody else's. So they want to develop their own numbers for their own site, rather than rely on being part of the generic Ontario vineyards. They want, no, I am not the same as the Beamsville bench. I am not the same as the St. David's bench. I am not the same as Queenston. I am not the same as Creek Shores. In fact, I could be in Creek Shores, which is a very large sub-appellation, and I could have a block on one side of the road, and across the road the soil type changes, and I will have very different numbers. And I have to be that specific, site specific. So it's really important. So, what do you have available? That's a soil test. What's the vine taking up? That's your tissue test. What can you change? This is where you look at fertilization with amendments. Organic, synthetic, commercial fertilizers, whichever you want. Well, the first thing that I tell everybody to do, and I've done this at more places now than start digging some pits and find out where your roots are. In many cases, poor uptake of nutrients is due to confined root systems. People think they're going down to a greater depth than they are. They don't know where their roots are actually located. What do? Start with a good map. I always tell people, this is a new winery operation that's going to be starting in Niagara. They had some existing blocks in places. Works better if I order. But they wanted to start with this new area with planning it out with Pinot Noir and Chardonnay. Now this area is only 4.5 acres in size. So we said, okay, let's get some representative information out of here. Well, when I went over there, you can see I wound up digging 1, 2, 3, 4, 5, 6 pits in that small area because there was a lot of topographic variance, and I needed to know what was there. Plus, we wanted to know historically around there what was going on for future development to see whether there was differentiation. Now, for a small site that's only about 15 acres total, to dig 13 pits, that's a lot of digging. But you do it at the beginning, you do it once. Premium viticulture around the world starts with checking the ground really well. And it's something that we've, I would say, failed to do in Niagara. There was a lot of mass planting on lands that were never really fully checked out, and now issues are occurring. It's easier to fix something before you start than after the fact. It's kind of like build the right foundation for your house will go up square. Have a poor foundation for your house and then wonder why your walls and plaster crack later on. Well, you just didn't do your homework at the beginning. And understand, this was just a couple of the pits. This is pit 11, showing at an A horizon that was 12 inches, a B horizon down to 22 inches, and my C horizon below it. And these were just some of the things that we found out. There had been clover mix and barnyard grasses present in the area. Highly fractured, good tilth, quite friable. So this is the history that I'd found about it. Similar on that small piece of land, soil pit 13, suddenly now I only had 5 inches of the A horizon. Much shallower topsoil area. So this is going to restrict my vine growth and size. But as long as I'm aware of it at the beginning, then I can understand what I've got to try and correct. So this is why digging the holes and doing a good background check is really, really important. So what's a soil analysis give you? It'll give you the general composition of the soil. Understand that it's a snapshot. It will tell you the soil pH at the time you sampled it, and it can assist you for the future. This will allow you to do it in other ways. Where do you take soil samples and where should you take them? Personally, I always look at the soil sampling zone. What I want to look at in a commercial vineyard, I want to go where 90% of the roots are going to be. I'm going to sample beneath the vine, roughly 50 centimeters on each side of the vine. I am not as concerned about the mid-row sampling because very few of the uptake roots that I'm thinking about are going to be located there. The bulk of your nutrient-taking roots are going to be in this area. So why not find out what's there? Because if I'm going to add things, that's where it's going to be of most importance. Now, depending on how you want to do it, you can do it topographically. In other words, you can isolate based on slopes, low areas, whether you've got sand ridges, or if you want, you can do grid sampling methods. Everybody has a different way of kind of trying to do things. Personally, I'm more of a fan of the topographic as best I can to give you a reading. But when I show you I've got one of Jim's wonderful slides, a couple of Jim's slides in here showing spatial variability of some of the nutrients, then you have to say, how am I going to treat this? Well, that's a whole other game. I haven't got the solid answers for you on this one yet. The other one that we've done, this is showing Ryan Brewster that works with me. We've been going out with our GPS unit and doing three-dimensional mapping of vineyards. This was that open land where those pits were. And I showed you, you remember pit 11 and pit 13 that had very different A horizon? That's where they were located on this map. But look at the sloping difference. There's a real drop off. There's almost like a small little ravine that went over here. And all along here, this was actually a stream. So your elevation changed from here back to here. But in 3D, you can get kind of an interesting sense. And what we found with people is we can pick out low spots in vineyards. And they will always be a problem area, regardless of your vineyard. Anywhere there's a low spot, even on a perfect slope, a low spot in there will always have issues. So if you can be aware of it at the beginning, then you might be adjusting your pruning and some of your nutrient work as well. The whole hope in the future, if we do get to more and more equipment that can fit us with precision agriculture, we might be able to do a lot more aggressive fertilizer work. But right now, we're pretty much stuck with the zone of best fit. Because once you start driving down the row with the wires and the posts, you can't turn around and go back. You got to go all the way to the end, and if you turn around and come back, nobody wants to drive up and down every row 5 or 6 times with different mixes. So we have to think about how we get past that. So the soil has done that. The tissue, again, gives us a general concentration at the time. It'll be variable at the time of season selected. That's why samples that are taken prior to bloom have very different results than those that are taken close to veraison. Maturity of the leaves, but also the rate of growth of the plant at that time and what its energy demands are. Nitrogen fluctuates over the whole season. I look at it to a limited degree, but just to give me a sense, but if I'm not seeing visible symptoms of nitrogen deficiency, rarely have you got a serious problem. Plant stress is not taken into consideration when you take the tissue samples unless you tell them at the lab you've been through a severe drought, or you've got a real high crop load, or you've done some recent pruning, or if you've got shading issues. All of those things are going to influence the nutrient level in that tissue because you're using the petiole of the leaf. But the lab doesn't take that into account. It says, no, you just gave me this stuff. I have no idea what stress you've been under prior to this, but I'll tell you what's in there. They do the absolute measurements. It does not tell you what's available in the soil. It just tells you what the plant has absorbed. If you've got a root problem and lack of absorption, you will have a tissue deficiency, yet your soil could still have excessive amounts. Now we got an issue. Could be water related. Where do we take them? If you're going to do it just before bloom, generally they take the leaf opposite the primary fruit cluster. It's critical that you take them and always look at them from the standpoint, and it has to be variety specific. Don't mix varieties. And if you've got different rootstocks in a block, don't mix the same variety that has different rootstocks. If you have a Chardonnay block on 3309 and a Chardonnay block on SO4, sample them separately. They will have different levels. If you're going to the Veraison, generally what I talk about is take petioles midway down that shoot, halfway from the tip, from the base to the tip, first mature leaf. Be very aware. The results will vary with the age of the vine. Young vines will give us lots of nitrogen, really aggressive growth, and be often deficient in some of the macros. Older vines are often in better balance. The cultivar you're choosing, whether it's Merlot, Cab Sauv, Cab Franc, Gewurz, you need to start to accumulate for yourself. When you took the sample, what part you took on it, and was it really representative of the area? Vine stress also has to be, you have to take into account. So when you interpret these things, you want to know whether is it due to a stress. And the other one is pesticide use. If people are spraying things like copper or sulfur, or there's some other ones, Mancozeb, which has manganese in it, you may have high levels of these particular elements show up in your results when in fact it's not a problem. It's a surface thing that just came off when they did the analysis. But again, the laboratory never asked you for all the pesticides that you may have used prior to. So it can skew their results. They just analyze what's there. This was just to give you an example of really what can happen talking about Merlot over looking at a tissue sample versus a soil sample. The home block here, it was adequate in 2011. Tissue was there. 2012, the tissue was adequate, but my soil, or the tissue here, Chardonnay versus Merlot, and the variabilities, even though the numbers are roughly comparable. The idea again is separating out by variety. The Chardonnay levels, quite high levels actually for potassium levels in the Chardonnay at 2.9. In Merlot, 4.94, 4.61. But for them, low would be around 3.5. Whereas on Chardonnay, that would be very high. Knowing where it has to be for particular varieties is critical. Or looking at if we try and compare soil and tissue, tissue sample in 2010. Soil, we never do a nitrogen because it's too highly variable. But when I look at it in 2010, I had potassium at 4.3 in this block. And when I looked at it in 2012, I had 3.5. My soil, though, was telling me that I was low. Even though in 2011, the soil level said low, my tissue, even in 2012, without correction, still was picking up. The plants were getting it. So we have to go back and take a look at, okay, was this really the available potassium, or what were the other factors in play at the time that we got that measurement? Or do we have just a poor representation of samples? Similarly, looking at a micronutrient like zinc, or even looking at boron. On a tissue at 42.1 and a tissue at 33, both adequate. In fact, anything for boron in tissue over 25 is quite fine. The laboratories and other ones try and push you to get up to 75 or actually almost 100. I'm not a fan of that. I've seen where they've recommended to get 60 to 65. That's way too much. In the soil, they're pushing for 1 to 1.5, way too high. 0.5 is more than adequate. More than adequate for grapes, especially under the moisture conditions we're in. If you're bone dry, maybe not. And again, another one looking at potassium, trying to look at just one end of a block to another end of a block. And there is variability, and that's one of the things that you'll have, and you got to live with. So be aware that the labs use generalizations. It's kind of like trying to get your dietary regime for yourself strictly by going to a newspaper. And I have the great fun of the one. It's kind of like saying, well, am I fit? Am I overweight? Am I obese? And you start using the BMI calculator, and that's still all over the map. There are some things that you just can't generalize. And I'm trying to get you to really think about your vineyard. You should be looking at each vineyard as an entity unto itself, in a block by block basis. How do you want to treat it? Soil results, as I said, they can vary with topography, cultivation, time of the year, prior application, and representativeness of the sample area and pesticide use. This is, as I said, Jim sent across to me, just showing the variability of potassium within a single block. Huge variability. Then when we overlay and take a look at it and suddenly throw in organic matter, we get this other completely different mapping of it. What you have is my way of saying, in your vineyard you have goulash. You have everything there. And at the end, you want the vineyard to perform by taking and making sure it's all in proper concert and balance. So you've got numbers. The general assumption is that 50% of the nutrients taken up by the vine get tied up. Roots, leaves, shoots. Crop removal is estimated at these levels. Roughly for every ton of grapes you take out, you're going to take out about 4.5 pounds of nitrogen, or roughly 2.5 or 2.25 kilos. Similarly, potassium, you're going to take out around 7 pounds or 3.5 kilos. That's what's physically going to be removed off site. So where does this come into play? If you get your vineyard into a steady state, you have to do the infamous Elton John circle of life. Whatever you take out, you better put back. And generally we have to put back a little bit more because we often lose some through leaching and groundwater and everything else. But that's important to remember when you get to these levels. But the amounts, it was amazing to look at, like 6 grams of boron. That's not very much boron. Yet people are telling you to put huge amounts of boron out into fertilizer applications. So, use local data and experience. The reason I say that, I can go to a book and I can pull out data from Ontario, the Northeast US, Australia, and Oregon. Boron in Ontario, according to Bob Klein from years ago, was 20 to 60 parts per million. The Northeast US says 25 to 50. Well, that's pretty close. Australia is anywhere from 30 to 100. And Oregon was 25 to 50. This is a big range. A big range. So if you were sending your stuff to Australia for interpretation, they may come back and tell you, load it up. But you don't need it. You really don't need it under our conditions. Iron, manganese. This one was 20 to 200. The Northeast is 100 to 1,500. Massive, massive differences. Zinc, again, same thing. And I just want to highlight to you, this is the variability, because the laboratories from these areas are going to use these base numbers to try and come up with recommendations. Okay. Test values for tissue, again, same thing. Australia goes from 2.2 to 4% for tissue. They like lush, big foliage. I don't know why, because it's a high water demand, but they do. Phosphorus, potassium. So again, I've just kind of summarized them just to show you what happens when you start to dig in, because people say, well, what numbers do I use? I come back to local. Develop your own numbers. Here's stuff on pH, anywhere from there, I was talking around 6.5. Phosphorus around 20 parts per million. Oregon, they can go up as high as 100 parts per million. Potassium, 120. Can be as low as 75 in the Northeast U.S. Again, varieties will all have different impacts on what the soil test should be at in the minor elements. Boron at 0.5. The U.S., they go up as high as 2 parts per million. That's really high levels. I'd be nervous on that. My comment here, this is a picture actually taken back in the early 1900s. This is the Niagara Falls concert band. The city of Niagara Falls actually had its own marching band, and people from Niagara and the Lake and Rest played. You got to remember the working concert. No single element performs alone. You have to remember you can't solve it by fixing one element. You have to take all of them in play. So what I'm going to do, I'm going to finish off with the top 10. Moved along pretty good. I'll take any questions right now, and then a comment that was made to me about giving presentations. Always finish when you talk, not based on the questions, because then they'll walk away with what you want them to remember. So, are there any questions? Art.
When does the vine have the maximum uptake or demand for nitrogen?
There's actually a couple phases. There's the peak development occurs about a week before bloom, and it peaks out just after bloom in terms of uptake of nitrogen through there. Then it'll drop off, and then there's actually a midsummer uptake of nitrogen in, I would say, mid-July. And then it declines at that point. And where this has come into play, and probably from the winemaking, is looking at yeast-assimilable nitrogen, where people have had YAN or the problems with stuck ferments. Some really good work has been done by Tim Martins and the others, and actually they found a way to, if you've been having troubles with it on drought years, their way of correcting it has been to use, believe it or not, a foliar urea spray, very close to Veraison, without impacting or producing late season vegetative growth. So this is again knowing, but that's the key periods for nitrogen.
Can I just get you to repeat any of the questions before I answer them? It's
okay.
Online here as well.
And sorry, the question previously was about when is the peak period for nitrogen uptake, and that's what I was talking about. Really, in the up to through the bloom period, mid-July, and then how you can correct yeast-assimilable nitrogen issues.
I was wondering if you had an opinion on the role of mycorrhizae forming associations with the plant roots to mobilize certain nutrients like phosphorus.
So my opinion on mycorrhizal associations with the roots of grapes and how they influence nutrient uptake.
Do you think it's just negligible influence? Or?
Do I think it's negligible? No, I think it actually has a significant influence. In fact, most plant species, once they've established in a particular place, there will be a natural development of mycorrhizae associated with their roots that are host-specific. Grapes have certain types of mycorrhizae that actually associate with them. So if you plant grapevines into a brand new piece of soil that's never seen a grapevine before, the buildup of these mycorrhizae is much slower, and it's more delayed. Whereas if they had been present at the early phase, when your root system is very small, mycorrhizae expand your surface area of the roots dramatically. So on a young grapevine, mycorrhizae have a massive influence, but on an older grapevine, not as much, because it already has enough fine fibrous root hairs there to make it present. So what they're doing is increasing. So yes, it does, but more so in the establishment phase, and probably I would say in the first 10 years of the grapevine's life, far more influence than later on. But the problem is it takes a few years for them to establish. So what some people have done is they've looked at mycorrhizal inoculations on grapevines at the time of planting to try and soak the vine. So some of them are already on those young vine roots to start. So when the plant's first starting, it's starting at the same time instead of having this time lag and delay.
According to our soil texture, how about the wine vigor, clayish or sandy soils? Which one is more vigorous?
Definitely more vigor in a sandier soil. Part of the reason is that it allows, and the question was clay versus sand for vigor on a grapevine. Because the vines can penetrate very quickly, there's access to nutrients very quickly in a sand soil. There's less impediment, less compaction, less tendency to develop hard pans. That being said, clays have better or higher capacity of holding nutrients in the soil because of greater surface area. Where we have to work with clay is to make sure that the mineral elements are made available to the plant. So that's why we have to look at drainage to eliminate excess water. Clays have a tendency to saturate and bog. We have to make sure along with it that there's lots of air in a clay soil because we can get a situation where the air can be locked away and unavailable for the roots. The third part is when clay's dry, they will dry like a piece of brick or concrete. So a root trying to penetrate or grow through it won't do it. Whereas sands are more fractured and will move through. So that's why initially grapevines will grow much more quickly on a sandy soil, but sandy soils can be less overall, have less nutrient available to it because it's a series of big beach ball particles instead of little pieces. So generally we'll see bigger vines on sandy loam soils, the classic peach grounds, and that than we will find on a clay soil. But a well-managed clay soil will still give you a good vine. A clay loam. Generally, we rarely do we go into straight clay. We're often in clay loam soils.
Earlier we were talking about digging soil pits and other roots that confined, or could have a confined that do like to water table or hard pan or more to vine spacing, or what's that?
The question was in digging of pits and finding confined roots, what was it due to? Primarily it's been due to compaction. In other words, a hard pen layer has been developed. In other locations where I've been, it has been due to very early poor drainage, which has led to saturated soil, so we have an elevated water table during the peak period of root development. But I have never seen in a good soil that's well fractured, well drained, any kind of reduced root growth on grapevines because they're closer together. In fact, the key work that's being done on that right now, Tony Wolf and Tremaine Hatch down at Virginia Tech, are planting grapevines in baskets, in confined nettings to try and keep the roots from getting out to help contain the vine size. Because the soil is so good that they know the roots will go. They know they put vines close together and they still get big, excessively large. So they have to find a way to confine the root system. So that to me looked like a very interesting project where if you had tile drainage and the rest, so there's lots of work going on. We're all spending a lot of time trying to figure out how to manage vine size.
Is it good to have a very high organic matter soil? Mine's about 4%.
Is it good to have a high organic matter level? Generally, your organic matter level, the highest that I find reasonably acceptable is around 4%. If we get above 4 to 5%, we often have excessive amounts of late season nitrogen release, which leads to late growth on the plant, which leads to reduced winter hardiness. If we get much lower on a lot of the sandy soils, the highest level we can get to is about 2.5 to 3%. If it gets down below 2% or 1.5%, that's not very good because it really impacts on microbial activity in the soil and it impacts on water holding capacity of the soil and the structure. So our targets, we try and get between 2.5% and 4%. Once we go above 4, we're into a luxury situation. Once we're below 2.5, we're in a deficiency situation. Any other questions? If not, I'll finish off with what, as I said, what I'm calling my top 10. They're not humorous by any stretch, but try and come up with them. Make a good site map. If you're going to walk away from here, make a map of your property. Your property will not grow. Your property will not shrink. Do it once. Do it by GPS, and you're easy. Collect data for your site over time. No two locations are the same. Know what block. Always keep a record on the block. Keep all identification of that block and number it in a fashion that you can always go by the same name. Now, if you get into a big business where you're buying farms, number your farms. Don't name them. A very good friend of mine, when they refer to the farm, they always refer to it with the name of the previous owner. An example would be the service farm that was owned by so-and-so. Well, no, it's not that, because once they sell it, the name changes again. Give it a number. Give it a record. It's easier to fight. I was on the 100 farm in block 101 than it is to say a person's name. Very simple way to do it. And much easier if you use Excel for filing things too. Compare your results with the on-site observations of performance. Look at the patients. Get out of your truck, get off of your tractor, and walk and look at stuff. Sample from good and poor areas on the same site to develop your own target values. Find your best plants, find the best performance, find the best, whatever criteria you're using, get those. Those are your base numbers, those are your target numbers. Don't rely on the lab to tell you what your targets are. The lab will tell you what they actually found. Now you need to say, are those proper targets or not? Be consistent. Look at a 5-year plan. Nobody wants to spend all their money sampling all their blocks for soil and tissue 1 year and then not do it for 3 years. If you break it out over time sequentially, sample a little bit every year can help you than it is to try and sample everything all in 1 year. Make sure you've got a real need. If you're not seeing visible symptoms, you probably haven't got a high need for it. If you haven't got poor quality fruit, if you haven't got a lot of things, you might be on the cusp of running into issues, but that generally doesn't need the rates that are often recommended back to you. Nutrient applications are not cheap. And the other one is if you start to play the blending game, which your suppliers will often do, oh, I can mix that with that. I talked to one of the places, they often result in double the price of the materials because they're now the minute they start to blend with them. So start to look at what's already commercially available and figure out how to utilize it. Will be cheaper per dollar unit when you apply it. Foliar fertilizers are great. I'm not against them, but you only use them when you've got visible symptoms. Because if you're just throwing it out there because you think you might need it, you're spending money. Oh, well, it doesn't matter to me. It's only $4 or $5 an acre. Well, if you've got 300 acres, $4 or $5, $1,500. A couple of times, it's $6,000. Now, in the grand scheme, not much. Yeah, it is. They all add up. Every time you throw something you don't need out there, you're wasting money. All-purpose foliar products, unless you've got an all-purpose deficiency, throwing all-purpose out there may not be your best answer. If you've got 2 or 3 or 4 things that you can visibly see that are going on with the vine, yeah, that's probably a good choice. But generally otherwise, no, you're better off bang for your buck, go back to a product that you really need and look at it and do a cost breakdown. And finally, the last one, walk your vineyard regularly. A lot of people over time, they become managers, they forget to be doers. And the doers are the ones that are out in the field seeing it because you have an employee come in and say, you realize in this corner, in this block, there's a bad section? And you haven't seen it because you haven't been there in 6 weeks or 8 weeks, really? Because you're just flying around doing everything else, which is part of it. But you need to take time to periodically get on either in a gator or get on an ATV and drive the blocks. Take a look. Get around to different spots. The other part is use your own experience. Use your neighbor's experience. Don't rely strictly on a chemical salesman or a fertilizer salesman or even go into the desk where you buy your inputs. Their business is to keep you in business, but their business is also to sell you something. That's why I spend a lot of time, no, come back to, do you really need it or not? Use your senses. How does it look? Do they feel right when you touch them? If you taste it. Does it taste correctly for the fruit? And does it smell right? Have you got issues with rots and other things that are being traced back? It can tell you a lot more than trying to find something from a piece of paper. This is where I dig out a lot of my information. These are the most recent ones. If I was going to direct any of you to one particular book that I would recommend for any viticulture student or any grower for their library to tell you a lot, The Science of Grapevine's Anatomy and Physiology by Marcus Keller out of Washington State. It was published in 2010. It has a ton of information for you. Yeah, there's a lot of science in it, but when you go read through it, you'll find little tidbits of information. That's where I learned about magnesium and the whole issue that it's involved with sugar and starch movement out of leaves. The papers cited in it. Oh, okay, now this starts to make more sense. Little bits you'll learn every time. With that, this is a picture of Joseph Cromie Vineyard and Winery taken when I was in Launceston, Tasmania last year at the Cool Climate Conference. So when we were down there, they had massive heat this year, but the temperatures there was about 26 degrees Celsius at this time of year. So now you can understand why I sometimes you wish you were back where you were a year ago. Thank you.
Thank you very much, Kevin. That was a very interesting talk, a lot of good advice, and it was a really good review for myself as well. A lot of good information there. So thank you very much for joining us today. The next COVI lecture series will be in 2 weeks' time, and it's Dr. George Vandermeer from University of Guelph, and he's going to be talking about getting started in wine fermentations, challenges the yeast has to overcome. So please join us again in a couple weeks' time, and thank you for your attention today.